Good afternoon to everyone, and thank you for registering to this live webinar. A special thanks to those listening from Americas and Asia, despite the too early or, on the contrary, the too late hours. My name is Agneta Kish from the Institute of Analytical Sciences in Lyon, France, and I will be hosting this webinar on behalf of the Early Career Members Network. This is actually the fourth of our webinar series, and it is brought to you by the Early Career Members Network Committee. This session, as you may already know, is free for all the attendants, and I would like to inform you that all the material will be recorded and uploaded to the Metabolomics Society website, allowing access to live content at your own convenience. Please connect with the Metabolomics Society on Facebook, Twitter, and LinkedIn. Also, we would like to have your feedback about the Early Career Members Network, and you can share your ideas and suggestions to the email address info.emn@metabolomicsociety.org. Before introducing our speaker, I would like to remind you that the International Conference of the Metabolomics Society, Metabolomics 2015, will be held in San Francisco at the end of June, and the EMN committee is hosting a welcome reception on the 28th of June and 4 workshops on the 29th of June. I will be giving some details about these 4 workshops a little bit later at the end of the webinar. Today's speaker, Dr. Christophe Junot, is the head of a drug metabolism studies laboratory at the French Atomic Energy and Alternative Energies Commission. Dr. Junot is a member of the board of the French Metabolomics and Fluoxomics Network, RFMF, and the deputy coordinator of the French Metabo Hub platform, where he is also in charge of the coordination of analytical chemistry development. There will be time after the webinar for questions, so please send your questions by using the questions option of your webinar application. We can type the question throughout the presentation, and our speaker will answer them live at the end of the webinar. I think we are ready to start, and I will pass on to our webinar speaker, Dr. Christophe Junot.
Okay, thank you, Agneta, for the introduction. And first of all, I'd like to thank the Early Career Members Network of the Metabolomics Society for the invitation. It's a pleasure to be here with you. So the title of my talk will address high-resolution mass spectrometry, and I would like to show you how to start in the field of analytical chemistry and go until biomarker discovery. But I would like to. Oh, I have a problem. With the presentation. Okay, so I'd like to start with a presentation of the lab I'm in charge of. This is the Drug Metabolism Research Laboratory, LEM, and most of it includes 30 people. I would say 2/3 of them are involved in mass spectrometry for biological media. So quantification of small molecules for DMPK purposes, quantification of proteins for DMPK, but also for biomarker, protein biomarker quantification, structural analysis of protein, and of course, omics-based analysis, such as metabolomics, lipidomics, and glycomics. And we are also involved in cellular pharmacology and antibody engineering, but I won't develop these two aspects. So we recently launched 5 years ago a small company named Profilomic, which develops metabolomics for biomarker discovery and for environmental sciences. And our lab is also involved in Metabo Hub, which is a French national metabolomic infrastructure, which regroups four platforms in Saclay, Clermont-Ferrand, Bordeaux, and Toulouse. So we are an analytical chemistry laboratory And we started in the field of metabolomics more than 10 years ago by developing and validating analytical methods for metabolite profiling and also metabolite quantification in close collaboration with physicians, biologists, and also clinicians. In the field of genetic diseases of red blood cells, for example, hepatic encephalopathy, antibiotic resistance, and also non-encephalopathies. And in the course of all these applications, we had also the opportunity to develop some spectral database, and I will specifically address this point. So our basis of our work is mass spectrometry, and more precisely, high-resolution mass spectrometry. So high-resolution mass spectrometry enables to detect more metabolites and also to improve their identification thanks to two physical properties that are mass resolution. That's to say the ability to discriminate between adjacent peaks. You have an illustration here. This is a mass spectra from a lipidomic experiment from a cerebrospinal fluid sample, CSF. So you see here that the main species is phosphatidylinosytol, and there is also the possibility to detect sulfated compound, but this ion is really close to the C13 peak of the phosphatidylinosytol, and you see here that without high resolution and even ultra high resolution mass spectrometry, this would have been impossible to detect these species. So we really believe that high resolution mass spectrometry is a great improvement for metabolite detection. And there's also the second property, which is mass accuracy, which enables to obtain elemental composition that makes database query really more simple. But so we for a long time we believed that it was sufficient, but actually it is not, and we realized that a few years ago by performing annotation of human biological fluids such as urines. And as you can see here on the top of the figure on your right, this is a total. This is an extract, a chromatographic peak obtained on a C18 column from a urine sample. So first of all, this metabolite is not retained because it's eluted in the death volume, and it is supposed to correspond to methyl nicotinamide, and we checked that because we have this compound in our chemical library. Okay, but when moving to another column, which is a PFPP column, you can see that you don't have one peak anymore, but two, which correspond to two isomers of this metabolite. So, having that in mind, we began to develop some. We went back to chromatographic development, and so we now used three different kinds of column with complementary selectivity. First, a typical reverse phase column, which is a C8 or C18 column. Also a PFPP column, which enables to detect amines and also aromatic compounds. And at last, a helix system, which is really efficient regarding organic acids and also sugars, so highly polar compounds. And this is coupled to high-resolution mass spectrometry, such as Orbitrap or also QTOF instruments. We evaluated these three systems on our chemical libraries. So we have a chemical library of more than 1,000 compounds, and we selected a little bit more than 600 reference compounds. And as you can see here, by using you have here the metabolites that are retained on the colon, not detected by the mass spectrometer only. So you can see here that we are able to retain efficiently a little bit more than 200 metabolites of the 600. And if we are adding the two other columns, we obtain a really nice complementarity. And actually, if we could work only with two systems, the Helic one operated in the with a negative one detection, and the PFPP one with a positive ion mode detection, and we would be close to the exhaustivity regarding these 600 compounds. So now some quick some examples in order to demonstrate the added values of such an approach. First of all, isobaric compounds such as hypoxanthine and threonic acid. These two compounds are both of them are eluted in the near the depth volume on the reverse phase system, as you can see here. The hypoxanthine is the most intense compound, and unfortunately we only managed to get MSMS information on this compound, and so we were not able to confirm the identity of threonic acid, and this was achieved by analyzing the same sample on the ZigP Helic colon. Here you can see that we are able to confidently separate these two compounds, and we are able to obtain the MSMS spectrum of threonic acid. Another example regarding isomers this time. Actually, the MSMS spectra may not be the good solution sometimes, and this is here the case with these isomers of dihydrobenzoic acid. Actually, both isomers are detected in plasma, and they have the same MSMS spectrum, so loss of water and CO2. And the only way to discriminate between these two compounds is to analyze them on actually on the PFPP system. So MSMS is often not the good solution, and chromatography also remains a key technology, of course. That's our conclusion. So now, just to finish regarding analytical chemistry that we develop at the laboratory, and to move to some biological applications, we are involved in the development of a spectral database since 2007 or 8. And why do we do that? We do that because we know that although electrospray mass spectrometry is a soft ionization technique, it generates several ions from a single molecule during the electronebulization process. And you have here the example on the top of this figure. So you start by a mixture of compounds that can be partially separated thanks to the LC system. And if we are focusing on the yellow molecule, you see that during the electronebulization process, one can generate pseudomolecular ions, but also fragment ions, adducts, and actually all these ions are present in the peak table obtained from the software for automatic peak detection and alignment, and so this may lead to erratic annotations, and this is exemplified here. So as you can see here, you have here an extract of a peak table with a M2D ratio, retention time formula, compound identification, and also annotations. So the first level of annotation deals with public databases. So you are just matching an accurate measured mass to a theoretical one contained in a public database such as HMDB, KEGG, or Metlin. And if you do that, you have many annotation possibilities. And you see here that, for example, the mass, the first line at 188 could correspond to diethylatrazine or 3-amino-2-naphthoic acid, but actually it is only a loss. It is actually only a loss of ammonia from a tryptophan molecule. So this is the reason why we are really involved in the building of such spectral databases, because it helps to make biological sense for the interpretation of biological experiments. Okay, so by this means we run some annotation of biological matrices programs, plasma, urine, red blood cells, and also cerebrospinal fluid. And so we managed to identify or to carefully annotate with chromatographic retention times, accurate measured mass, and also MSMS spectra, a few hundreds of metabolites in these matrices. And for us, the take-home message is that there are at least 30% of isomers in these biofluids. So this is this really emphasizes the need of chromatography and of a good chromatography, and mass spectrometry and high-resolution mass spectrometry is for sure not sufficient. And of course, this work will be amplified within the MetaboHub program. We are also involved in lipidomics. I unfortunately don't have time to detail that, but we are able to annotate, to confidently annotate with retention time and MSMS spectra few hundreds of lipid species in plasma, cerebrospinal fluid, but also mammalian cell extracts. And we are also involved in glycomics, which is at the interface of metabolomics, lipidomics, and proteomics, because we are detecting free and also bound glycans with MALDI-TOF, MALDI-TOF, and also LCMS technologies. So this was an overview of what we are doing in our laboratory, and now I'd like to continue with two short examples. The first one deals with the analysis of cerebrospinal fluid metabolome for the study of inborn errors of metabolism. This work is made in collaboration with PTA-Salpêtrière Hospital, and so we are focusing on inborn errors of metabolism. What are these diseases? Actually, they correspond to genetic. They are genetic diseases, and they correspond to a mutation or of an enzyme or of a gene, sorry, that codes for an enzyme or a transport protein. And contrary to neurodegenerative diseases, IEM are prone to treatment. So you can, by a protein substitution, for example, by diet or inhibitors. And actually, when the enzyme or the transport protein doesn't work anymore, you have an accumulation of the substrate. So you can modify that by diet intervention or supply in order to compensate for the loss of B, or shunt activation of residual enzyme activity. And why are we interested in that? Because actually only 500 IEM are identified, and by knowing that more than 3,000 genes are codes for protein, for enzyme or transport protein, many of them could be could lead, many of these mutations on these genes could lead to IEM, and so there are many IEM that are still unknown. And this is relatively well known in newborn and children, but far less in adults. And in the PTA-Salpêtrière Hospital, the neurologists have a cohort of a little bit less than 600 patients that are referred for suspicion of metabolic diseases. And as you can see here, 15% of this cohort correspond to real inborn or diagnosed inborn error of metabolism, and a little bit less than 70% correspond to unknown encephalopathies. So some of these patients may have some inborn error of metabolism, and our aim is to stratify this patient in order to highlight new diseases. So you can see here a typical dendrogram from a hierarchical cluster analysis of human CSF. This has been obtained on a reverse-phase system coupled to an exhaustive mass spectrometer. So you can see that the situation is complex, but by looking a little bit more in depth, we managed actually to cluster some patients. The first one highlighted in red correspond to a portosystemic shunt that I will address a little bit later, and all these samples highlighted in green. Actually, we realized that four of them correspond to a neurological syndrome named CAFSA. This CAFSA syndrome has been recently described in 2009, and it corresponds to cerebellar ataxia, which is characterized by an accumulation of free sialic acid from NMR spectroscopy analysis. And so the colleagues from the PTS-Salpêtrière highlighted 4 gene candidates, but nothing really emerged. And so by analyzing this the metabotypes of these patients, actually we realized that there are many other metabolites as sialic acid that are increased, whose plasma, whose CSF, for example, concentration are increased, such as carbohydrate, hydroxy acids, and so on. And interestingly, we also demonstrate that these four metabotypes were really closed on a metabolomic point of view to a patient corresponding to a patient who has a Kern-Serre syndrome, which is a genetic disease with a massive mitochondrial DNA deletion. So this path, this enables for the clinician to have new hypotheses, and so this metabolic fingerprints enabled could be of help to analyze exome sequencing data and to provide new biological hypotheses. Now we go back to this patient having a portosystemic shunt. Actually, a portosystemic shunt leads to a hepatic encephalopathy, and I will explain you that now. And so we decided to study more in depth this pathology, and so still in collaboration with PTS-Salpêtrière, but with another group, Dominique Tabu and Nicolas Weiss, we decided to evaluate, to perform a metabolomic analysis of CSF samples obtained from a patient having hepatic encephalopathy. So hepatic encephalopathy is a real issue of critical care medicine, and it's a complication of it's a neurological complication of acute or chronic liver disease. Why? Because actually ammonia is usually metabolized in the liver, and when the liver doesn't work anymore, you have increased plasma concentration of ammonia that can go everywhere else. So, for example, in the brain or in the kidney and so on. And so this high concentration of ammonia is toxic. And so this is important because actually 60 to 80% of cirrhotic patients may have hepatic encephalopathy, but the diagnosis has really to be improved. So the pathophysiological mechanism of hepatic encephalopathy is still poorly understood, because there's hyperammonemia, but this does not explain everything. There's also inflammation and also altered permeability of the blood-brain barrier. And so we decided to use metabolomics for patient stratification, and also to know whether or not we could highlight some pharmacological targets for the treatment of these patients. So this is a summary, because I will address that at San Francisco at the end of this month at the Metabolomics Society conference. But you can see here from a score plot of principal component analysis that it is really easy to discriminate between hepatic encephalopathy patients in red and control patient in blue. And I don't have really the time to go more in depth, but we obtained several tens of discriminating metabolites that belong to various chemical families, such as amino acids and derivatives, sugars, organic acids, acylcarnitines, and so on. And this metabolomic study led us to propose a hypothesis regarding the alteration of energy metabolism in CSF of these patients. So we know from previous study performed in animal models that Krebs cycle activity maybe is reduced in hepatic encephalopathy. And interestingly, we are all the Krebs cycle intermediates that we are able to detect. That's to say, all the metabolites that are not highlighted in gray color are not altered in hepatic encephalopathy patients. You see here the levels are unchanged. But just around this cycle, we observe many metabolic perturbations. So, of course, highly increased glutamate and glutamine levels, which are the consequence of hyperammonemia. And our hypothesis is that the Krebs cycle is impaired because some of its intermediates are involved in ammonia detoxification by the synthesis of glutamate and glutamine, as you can see here. For example, the level of lysines are decreased because the color is in blue, because lysine could be depleted because of its involvement in glutamate synthesis. And interestingly, we also observed an accumulation of pyruvic acid, of acylcarnitines, and also of acetyl amino acids. And we think our hypothesis is that there is an accumulation of acetyl-CoA, which is not, or which is no more or far less involved in Krebs cycle, and so this accumulation of acetyl-CoA could lead to acetylation of amino acids or other compounds such as nucleosides, and this could be a good biomarker of Krebs cycle dysfunction. So by performing this study, we demonstrated that that energy metabolism, or we suggested, sorry, that energy metabolism could be an appropriate pharmacological target to treat such patients. Okay, so in conclusion, the future of our laboratory will be the analysis of medical cohorts. Of course, we are already involved in this field, and this will be achieved through the MetaboHub program, of course, by increasing our metabolite identification and annotation capability, by also developing tools for data fusion in collaboration with colleagues of CEA, the group of Etienne Thévenot, and also by working on metabolic networks in collaboration with Fabien Jourdan in Toulouse. And so I would like to thank all my colleagues for this work, people from all the people, the postdoctorand, the PhD of the laboratory of Paris 6 University also, of people from the CEL list who are involved in bioinformatics, and also people with a profilomic company with whom we collaborate, and all my colleagues of Metabolomics. And thank you for your attention.
Thank you, Christophe, for this very inspiring presentation and very complete, going from analytical chemistry developments to today's applications. We have now time for some questions. So for the attendees, please remember, if you haven't already, you can still type the questions through the questions option on your panel. I will ask them to. I will ask the questions to our presenter. So I will probably take the first question from an early career attendee. So the question is, what is, in your opinion, the bottleneck of the MS-based metabolomics field, and what advice would you give to early career scientists?
Okay, so there are unfortunately many bottlenecks. The first one is at the level of method development. So how to develop a metabolite, an appropriate metabolite extraction method, for example, how to validate such fingerprints. What I would advise is to start with few tens, perhaps ideally between 50 and 100 reference compounds that will be of help for method development, and that also will be of help to carefully evaluate the software for automatic detection and alignment and integration of features. So start with reference compounds and don't go immediately on biological samples. There are also few reference protocols that have been published in Nature protocols, and so these protocols could be good starting points. The second issue is at the level of this software for automatic peak detection, and so they are really depending on your instrument and also on the biological matrix you analyze. So the same as for method development. I advise to start with reference compound and with simple design of experiments, such as a triplicate analysis of a pool of standard compounds with serial dilutions. So by this means you will check the detection, feature detection, but also the integration, the peak integration capability of the software or your parameters. Regarding identification, the situation is far more improved than in the past, because manufacturers started to build some CID databases, and also there are more and more data on public databases such as MassBank. There are also tools such as MedFrag that helps for MSMS interpretation, and you can use this tool even if you are not an expert scientist, an expert analytical chemist. Of course, you have to use that with caution, but this is still available. And the last issue, which is perhaps the more difficult to address, is how to share such data, because there is a huge intra and inter-batch variability, and this really prevents data sharing. And there is also, you may know that mass spectrometers, new mass spectrometers are also released every 6 months. So the big challenge is to be able to analyze data that could have been obtained with old mass spectrometers, and to fusion this data with those obtained on more recent mass spectrometers. And this can be now achieved thanks to algorithms such as LOES, for example, that have been published 4 years ago. But and this could be addressed by using QC samples that can be analyzed during the experiment. But this is not an ideal solution because we may have some long-term stability issues that are really difficult to handle. And so, to my opinion, the future of that will be large-scale absolute quantification.
Okay, thank you. There is an attendee who is asking if the database that you mentioned is something that is local or public, and if there are any collaborative efforts on building databases.
Okay, so there are two categories of two kinds of databases. There are databases obtained on electrospray mass spectrometry experiments, so LCMS, for annotation of for peak table annotation, and there are also spectral databases that address MSMS, CID experiments. So the first category of database, so spectral database for compound annotation, can be shared because many ions are common to different kinds of experiments, and this within MetaboHub we now begin to share these databases by analyzing the same reference compounds in different chromatographic systems. So it's possible to share, to use them only with the mass spectrometry information, and also you can use the chromatographic information, but it is really more difficult. And regarding the second category, it's more difficult because there is a huge variability from one instrument to the other. So in many cases, it's all right, but the problem will come, for example, for the analysis of isomers.
Okay, thank you. Third question. Is there a limit for high resolution? Is the ultra high resolution of the latest instruments justified for metabolomic studies, or actually how much resolution do we need?
Whether or not there is a limit, actually I don't know. I just know that MS instruments are more and more powerful. And so regarding I would say regarding the need for metabolomics, I would say as resolutive as possible, actually, because biological matrices are really complex, and we really have to discriminate between ions that are isotopes and ions that could correspond to pseudomolecular ions. So the more we have, the more resolutive power we have, for sure the best it is.
Okay, thank you again. And what do you think about the role of the analysis made in negative and in positive mode? In many cases, you will get the same compound, and would it be more a problem than a help?
No, I think it's really complementary, and I really advise everybody to perform the analysis with both modes. And a good criteria, for example, for variable selection is to have the same trends, the same concentration trends in the data acquired in both modes. and that's what we are doing at lab. Actually, when we remove all features that do not exhibit the same trends in the negative and the positive mode. So they are really complementary actually.
Okay, thank you. There is one attendee who would like to know which internal standard you use in helic mode, in positive and in negative.
Sorry, I did not catch the beginning of the question.
What internal standard was used in helic mode in positive and negative ionization?
Okay, so we have a mixture of, if my memory is well, actually it is not well, more than 10, we have 10 xenobiotics or labeled metabolites. I don't have the complete composition of our pool in mind, but it is already published. So please refer to our publication. Actually, we don't have any standard mixtures dedicated to helic. We use this. I think there are 14. We use these standards in C8, C18, PFPP, and helic colons. And these standards are not used for normalization purposes, but only to check that the injection process happened well. So it's just the beginning of the analysis. We start by carefully checking the behavior of these standards in blank samples, and if it is not okay, we stop with biology. We stop with the interpretation and start the experiment again.
Okay. And another question from an attendee who just started using MetFrag. He's saying that the results were not reliable, even if he used fragmentation. And although such systems were improved, there are still some drawbacks. And in your opinion, how reliable is the replication?
Regarding MSMS experiments?
Yes. I think so, yes.
Regarding MSMS experiments, yes, for sure. I think these tools such as Mass Frontier, MetFrag, and are tools that could be relevant for beginners or for experienced scientists in order to save time. But they are not the Holy Bible, for sure. And so I think I don't have many solutions to this issue unless the implementation of a collaboration with people experienced in the field of CID spectra interpretation.
Okay.
But this is actually a good starting point, and if the metabolite is commercially available, this is the opportunity to purchase it without purchasing many others that will lead to erratic identification. So even if you are not experienced, it can give you information and it can drive the purchase of synthetic compounds. So I think it's a good point for that.
Okay, thank you. And one last question maybe. Could you give some examples of educational programs like masters and trainings adapted to the needs of the MS-based metabolomics field?
My answer, unfortunately, will be restricted to the country I know best. That's to say France. And so in France, the situation is simple. Actually, there are only few programs, and even no program actually to my knowledge. There is a Master 2 degree in Toulouse dedicated to both metabolomics and proteomics, but to my knowledge, this is the only one. But I think more and more the master degrees, the French master degree, include metabolomics in the courses, but these are only courses, and to my knowledge, there is not, for the moment, a metabolomic master that is specific to this field. And it's, I think it will come, but not for now. So the advice I could bring to all of you is to attend to a good master degree regarding mass spectrometry, to attend to an analytical chemistry master degree, and after to specialize in metabolomics, thanks to a training course or practical courses in the field of this master degree.
Okay, thank you very much for all these answers. We can maybe now come back to the presentation slides, because before we conclude our session today, I would like to quickly advertise the 11th International Conference of the Metabolomics Society that will be held in San Francisco between the 29th of June and the 2nd of July, sorry. As previously said, the EMN committee is hosting a welcome event and a reception on the 28th of June and 4 workshops on the 29th of June, that is on Monday. Early and regular registrations are closed, but you can still register late until the start of the conference. And as I promised earlier, I will give you a few details about the 4 workshops that the EMN organizes. The first workshop will be about big data, and we'll have Dr. Douglas Kell from University of Manchester and Rob Davidson from GigaScience as invited speakers. The second workshop will be about fluxomics, pathway analysis, and metabolic engineering with 4 speakers: Professor James Lyo from the University of California, Professor Akiko Kondo from Riken Institute for Sustainable Resources Sciences, and Professor Will Sauer from ETH Zurich. The third workshop will be about careers in metabolomics, and we will have speakers from academia, industry, and government. And there is also a workshop about research integrity, where we invited Professor Roy Goodacre from the University of Manchester and Dr. Philippe Roca-Serra from the University of Oxford. As I said earlier, please connect with the Metabolomics Society on Facebook, on Twitter, and LinkedIn, and feel free to share all your ideas and suggestions with us at the email address that you can see on the screen, info.emn@metabolomicsociety.org. And if you have any other questions, you can still send them to us by email. We will try to connect you with the speaker, and you will also find some answers in the recordings that will be made available after this live webinar. We will be having another webinar soon, and more details will be sent as soon as we have confirmed details about the speaker, about the exact date and time. Thank you for attending, and thank you, Christophe, once again for accepting to give this presentation. I would also like to thank everyone for their support in organizing the webinar, and I hope you all enjoyed today's presentation. Have a nice day today, and thank you again.